A new generation of science is emerging from Africa where innovation is engineered and solutions are delivered across education, biotechnology, healthcare, and technology. This is Genomark Holdings, and we are a multidisciplinary innovation company transforming lives through genomics, artificial intelligence, and education. Across our subsidiaries, we are translating research into impact. As Genomark Labs, we advance precision medicine. As geneious, we harness nature to create sustainable therapies. As Genomark Services and Consults, we empower institutions with innovative scientific services. At Genomark Innovation Hub, we build startups that redefine Africa's health future. And through the Genomark Institute Incorporated, we are driving education and research in genomics and bioinformatics. Today, I introduce GoGenBio, an initiative that brings genomics education directly to communities worldwide, equipping people with practical skills to innovate, build, and lead in today's biotech era. From Africa to the world, Genomark Holdings is forging a sustainable future powered by competence, innovation, and collaboration. Welcome to Genomark Holdings.
Hi, and welcome to the introduction of transcriptomics on Use Galaxy. So before we go into the hands-on section, we're going to go through the basic introduction of transcriptomics to lay a foundation on what transcriptomics is and to get us started, to help us understand what we're actually going to be doing. So firstly, what is transcriptomics? Transcriptomics is a study of a complete set of RNA transcripts produced in a cell, a tissue, or an organism. Now, when we say a complete set of RNA transcript, that is every RNA produced by an organism. We know that the RNA is produced from the DNA. The DNA is responsible for having every code that is in an organism, right? It's in the nucleus, and we know the DNA cannot move out of the nucleus. So the RNA is what moves out of the nucleus, carrying these codes or these messages that needs to be relayed to other parts of the cell or other parts of the organisms. And there are quite a number of RNAs that are produced. We have the messenger RNA, we have the ribosomal RNA, we have the transfer RNA, and we have the non-coding RNAs. All these RNAs have their specific sets of functions and what they are used for in the cell or organisms. Now, studying all these RNA produced and what they are used for is what transcriptomics is all about. Now, why is transcriptomics important? Why do we study transcriptomics? What's the relevance of studying and carrying out transcriptomics analysis? Firstly, we have understanding gene expressions. Like I said earlier, the DNA is what carries every code and that relates to an organism or an individual in general. But we can't directly study the DNA. So we study the RNA, which is the transcriptomics, to help us know what these genes are about, because the RNAs are what actually carries the message or the code outside of the nucleus. So by knowing what these RNAs are carrying, knowing the codes that they represent, we're able to understand how these genes are expressed, and we're also able to use it to something like a turn and off in different conditions. So in a particular set of conditions, because this set of RNA or this set of code are present, it means that this particular activity will occur. And when some set of RNAs or some set of code are not present, it also means that some set of activities are not going to occur. Also, we have disease research. In understanding the RNA produced by different organisms, we're able to understand how diseases alter the different functions of organisms when they are attacked by diseases. or when they are subjected to diseases condition. We have, like in diabetes, we are able to know how diabetes comes about by being able to understand the genes that are expressed at a particular time. And what are those genes? Those are the genes responsible for the production of insulin. When the genes responsible for the production of insulin is partially or totally silent, it means that insulin will not be produced. And when insulin is not produced, that will lead to diabetes. Also, there are some diseases or some pathogens that go to attack these cells' functions, resulting in these acts of functions not to be expressed. So when we're able to understand how all these diseases and pathogens are able to alter the normal functions of the cell, we're able to carry out researches on these diseases, which will enable us to know how to combat these diseases. Lastly, we have personalized medicine. What's personalized medicine about? Personalized medicine basically is creating drugs, treatments, therapies that are specific or tailored specifically to a particular individual based on their genes, based on how their genes are expressed. How does this work? Every individual, though all humans have the same, humans have 99% of their genes alike, but every individual has a way that a gene is expressed in them, which makes us unique. That's the variation that makes us humans. So there are some drugs or antibiotics or treatments or therapies that for a particular individual may work very fine, but for a different set of individual may have some types of reactions, some types of allergies or whatever that can occur by reason of those drugs. So by using transcriptomics and understanding how their genes are expressed, we're able to get drugs, able to get treatments that are specific to these individuals without having diverse or adverse reactions, negative reactions to the drugs. Nextly, we're going to be talking on how transcriptomics occurs or how it is done. And transcriptomics is done using a specific technique called RNA sequencing, which is a form of a next generation sequencing. And what is RNA sequencing about? RNA sequencing is just getting this RNA that we're talking from the organisms. And how is it done? Firstly, we have RNA extraction. This is collecting the RNA samples from a cell or an organism, and this is done in the lab. The RNA is extracted from the lab, and after that, we have something called library preparation. What this library preparation entails? Now, RNA on its own is a very volatile nucleotide base, meaning that when left alone, it easily disintegrates. Why is this? Now, the RNA and DNA can be likened to a pair of scissors. Now, the DNA is the complete set of scissors with its two arms completed. That is the DNA. Now, the RNA is just one arm of the scissors or the DNA. So, when RNA have been extracted from an organism, because they are just one pair, it is not the complete pair. It is easy for it to disintegrate or scatter. Let me use that word for us to be able to understand. It's easy for it to scatter or disintegrate. So what we do in library preparation is every nucleotide basis has a complementary pair. We have A to T and C to G. So for the RNA, what we do is we create a complementary pair for it to make it stable, and this is called a complementary DNA. When doing any analysis, wherever you see complementary DNA or cDNA, you know that that is an RNA that has been stabilized into a complementary DNA. Where the C is not present, you know that that is a complete DNA base. So after doing that, we have sequencing. Sequencing is, we have quite a number of sequencing techniques. We have the Illumina, we have the nanopods, but the most common is the Illumina. And what does this do is it takes these RNA strands and it reads each of the base pair that is present and helps us to know the arrangement of the base pair. So it's able, after doing sequencing, we're able to know that the arrangement is AT, CGT, CGA. So it helps us to read this RNA that has been extracted into a sequence we can understand. And also, what also is done during sequencing is that the RNA is a very long strand which contains millions of base pairs. On its own, it is difficult to read by the sequencer. So what is done is it cuts the RNA into short strands, and these short strands are called reads. So it cuts the RNA into short reads, which can be as low as 500 base pair to as high as 5,000 base pair. So after cutting this, it is easier for the sequencer to read this base pair and give us the sequence analysis. Now, after this is where we go into the data analysis, and this is where this is what we're actually going to be doing subsequently. Now, the data analysis has a lot of processes in it, and we'll be coming to that shortly. Now, application of transcriptomics. We can apply it in cancer research in finding the biomarkers. Every organism, when cancer occurs because a particular cell continues to grow without stopping. Now, in an organism or in humans, the cells have signals that tells them when to grow and when to stop growing. Now, the absence of this signal will cause these cells to grow rapidly without stop, and that's where cancer comes into play. So by being able to know what is responsible, the biomarkers are responsible for sending this signal, and knowing if these markers are actually activated or not, we're able to understand why this cancer has occurred and able to combat it. Nextly, we have infectious disease, the pathogen or virus to host interaction. What does transcriptomics help us here is in both ways. Using transcriptomics, we are able to know how these viruses or pathogens attacks the host defenses, the mechanisms they put in place to attack the host defenses. And from the host, we are able to know how this host, the immune defenses, responds to this attack. Using transcriptomics, like I said, we're able to know how the pathogens or virus attacks the host defenses and how the host immune defenses responds. So it is like a war zone. The viruses or the pathogens attacks and the host defense, and we're able to use transcriptomics to understand the analysis or the strategy. Let me use that word. We're able to understand the strategy the virus and the pathogen uses, and we're also able to understand the strategy of the host. In drug discovery and personalized medicine, I've given an explanation on this. In personalized medicine, we're able to tailor specific personalized treatment for every individual based on how their genes are expressed. And for drug discovery, by being able to understand how diseases are expressed, how pathogens attacks, how pathogens react to different environmental factors and things like that, we're able to know the best set of drugs for each pathogen or each disease that is expressed. Now, let's say for cholera, by being able to understand how the pathogen Vibrio cholerae responds to certain environmental factors, to certain antimicrobial factors, certain antibiotics factors, we're able to know the best antibiotics or the best treatment to combat cholera. Same with every other disease. Malaria, by being able to understand how the pathogen responsible for malaria reacts to different antibiotics or anti-malaria drugs, we're able to improve the drugs that have been created for the treatment of these diseases. And lastly, we have AI and machine learning in transcriptomics. Now, the data gotten from RNA, as I earlier said, can be quite large. It can run into millions or billions of base pair. So AI and machine learning has helped us to create a database that stores this information so that every research that has been done can be stored here, and we assist in very other researches that are going to occur. Also, it helps gives a specific analysis towards this um research, a pattern, like helping to recognize a pattern. So if a researcher has started a research, he's able to go through past record and using machine learning to see the patterns in which different other researches have gone. So he's able to know that, oh, for this particular organism, it has been following this specific pattern. So this organism has been, let's use Vibro cholerae. Vibro cholerae has been susceptible to this particular antibiotics for the last 15 years. And on doing his or her own research, he found out that that particular antibiotics, the pathogen has developed an immunity to it. Because there is already a database and a pattern that has been created by the machine learning, he is able to know that there has been a change in response in the pathogen he or she is working on. Now, lastly, we're going to go to the analysis, transcriptomics analysis. What is the workflow responsible for that transcriptomics analysis follow? Firstly, we have the pre-processing stage. Now, this involves collecting your data, analyzing and checking how good this data is. Now, there are a lot of data sets on the website available, a lot of public data sets from NCBI and other public websites for data collection. Most of these data, some of these data may come with very low quality. Now, if you can remember, I said during sequencing, the sequencer gives like a report of how this base pair is positioned. So it gives A, T, C, G, T, C, C, G. But there are instances of errors where what the sequencer wrote or placed was not actually correct. So instead of a T, it placed a C in that position. These are all things that we check during quality control. Also, some reads, when it cuts this RNA into short reads, some of these reads are of low quality, meaning that they actually do not have any codes in them. Because in transcriptomics, the RNA, if you remember, I said there are lots of RNA, but the RNA that we majorly work with you in transcriptomics is the messenger RNA. And this is because the messenger RNA is actually what carries the codes in the body or in the organism. It's what carries the code. So this is the RNA or the transcript you can actually use to run transcriptomics analysis. And some of these RNAs may not carry some of them do not carry complete codes. So when the sequencer cuts it, some parts may carry the code and some parts are not actually carrying a code. When it cuts the read, it may cut the part with code and a part without a code. So what quality check also help us do is to know how good our reads are, so we can know if we're working with a good sample. Nextly, we have alignment. For every organism, there have been a reference genome. When we say a reference genome, that is everything about that organism has been every gene about that organism has already been written out and assembled down for researchers. So we download this reference genome and we're able to align this particular gene we're working with, this particular set of data we're working with, to know the how well the reads align to the reference genome. So it shows us which of these genes are actually carrying code and which of these genes are what each of these genes are actually responsible for. If it's actually responsible for protein production and what protein is it responsible for, all this is what it tells us when we map. Nextly, we have the quantification. Now, this counts the number of genes that are expressed. Now, each read carries a certain amount of base pair, and each base pair codes for a particular protein. Now, all these codes or genes, as we call them, we during quantification we count how many was actually expressed, how many are actually expressing themselves. which means how many of these genes actually responsible for coding for something. Is it responsible for coding for protein? Is it responsible for coding for insulin? Is it responsible for coding for secondary metabolites? We are able to count how many genes are expressed. And lastly, we have normalization and differential expression. What this does is it identifies. Now we know during research work there is a stage where you do the control and the treatment. In the control, this is okay, let me use diabetes for example. The control can be a normal rat, let's use rat, a normal rat, which has good production of insulin. While the treatment can be rats that have been induced to show diabetes. So what does differential expression do? It helps us to know which of these genes are actually expressed. Now, in the control, we know that in the control rat, insulin will be expressed, while in the treated rat, insulin will not be expressed. So during differential expression, we're able to know that, oh, this in this control, insulin was expressed, and in the treatment, we're able to know that insulin is not expressed. Now, this is called the up and down regulated gene. We're able to know that in this particular condition, this gene is expressed, up regulated, and in this particular condition, it is not expressed, down regulated. It could also be the reverse. It could be that the control is actually a diabetic rat and the treated is a non-diabetic rat. In such a situation, in the diabetic rat where insulin is not present or is not expressed, that is the upregulated, and in the treatment where insulin is expressed, that's the downregulated, meaning that control is always the upregulated and treatment is the downregulated. So during differential expression, we're able to know which of these genes actually follows upregulation and downregulation. Nextly, we're going to be going into the practical section, the hands-on section, and we're going to dive into all these processes that are just explained, and we are going to see more about that. Thank you.
Welcome to the hands-on section of transcriptomics on use galaxy. In this section, we're going to dive into how to do transcriptomics analysis on use galaxy. So we are going to dive into that right now. So to do transcriptomics, we're going to use the website called Use Galaxy. Now you can go to any of your browser, whichever browser you are actually using, and just type Use Galaxy. And for me, it's already showing because I use it a lot. So just click on it, log in. Now this is what it's going to show. You just click on it and it will load. Good. So this is the interface of the Use Galaxy. This is the interface. For those of you that are using it for the first time, if you look at this side, it will show you register or log in. For me, my name is already there because I've already logged in. And immediately you're able to register, Use Galaxy is going to give you a storage usage of 250 GB. This is every analysis you do will be stored using the storage. So first in our text analysis workflow, I showed you that the first step needed or used. The first step is data collection and preprocessing. And to do that, we're going to get data from NCBI. NCBI is the web server we use to get data for analysis. And how do we do that? Firstly, before you come to use Galaxy, you have to go to NCBI. And to do that, the particular type of NCBI we are actually using is NCBI geodatabase. That is where we are going to be getting our data from. So this is the page, NCBI geo. The full meaning of geo is gene expression omics box. So this is where we get the data set we want. We are actually going for the accession number. So the organism you want to work with the organism you want to work with will be used, will placed on this set bar. You just type the organism. For me, I am actually using Vibro colorway. This is the organism I am going to be using. So you click on search. So now it gives two, but we're actually going to be using data set because it's the data set that we actually need. So you just click on this number. When you click on it, good. So a whole lot of research work have popped up. I'm coming. Like, so you just choose which of this work you are going to work with. Now, what I like telling people who are using, except you have no choice, but if you have a choice, when you are selecting the data you want to work with, when you are selecting the data you want to work with, try as much as possible to choose a data set that is has a small, like small in number, because why? The bigger the data set, the longer your analysis is going to run on Geneious software, on Galaxy, I mean, sorry, on Use Galaxy. So what you check, this side, this side determines how fast your data will run. The size is is the determinant factor of how your data will run. So what do we do? How do we get samples? After clicking on the, let me go back so we can actually follow. So you look for the one you want. If you notice, you see a lot of them have different organisms. Like this, the organism is Escherichia coli, and I do not want to work with it. You see this is Vibro colorway, and there are others, Vibro colorway. You just search you can search, there are different ways to choose. You can search using the topic. You can read the experiment that is actually going on and see if it's in line with what you want to do, what you are actually interested in doing. And if you are interested in it, you can get the data from there. So let's start. So firstly, try as much as possible to read the experimental type, the summary, and the overview, so you can actually know what the experiment is doing and what they were talking about. This now, they are antibiotics targeting the whole specific polysaccharides of glycocolorate. Next, what do? You come here where they say samples. You click on more. Good. So it has. So all these were the samples that were used in this research. There were 20 samples that were used in this research. And if you notice, each of them comes with a pair. This is the control. These are the treated. This is the control for this. These are the treated. So you can just pick in pair, pick them. So I'm going to pick one control and one treated. I'll pick one control and one treated, so we can work with the pair. So I've clicked on the control. Now you just go down. Now this is the number we are actually looking for. This is the number we want. So what do? You copy it. You copy this number and you come to your Galaxy. Good. So on getting to Galaxy, you go to get data. You don't have to download it. Once you've gotten the SRA number, Use Galaxy is connected to the is connected to NCBI database, and it's able to just collect it immediately. Now, before I do that, I am going to create a new history. The reason why I'm creating a new history is because I have different works I've been working on. So I can know the particular one I'm working on. I'm going to create a new history. But when you open and you register your Galaxy for the first time, this history unnamed will just pop up for you. what you do is you name it. I advise naming it with the research you are using so it is easier for you to know. So I'm going to rename mine now as Global Outreach. Okay, so you click on save and mine has been renamed to Global Outreach. So we have copied the SRA number from NCBI and we come to get data. You see, download and extract reads in BAM, download and extract reads in FASTQ. As I said in the first video, the data we work with for transcriptomics is the FASTQ data set. So just click on this. So this is the ascent is showing you SRA, the type of data set you are going to use is SRR ascension or SRA as it's called here. So I'm just going to save the one I copied. Save the one I copied. Okay. So sorry, there was an error. We made a mistake. Let's go back to the NCBI. You know, I said this SRA, this was the one we copied. But actually, this is not what we need. We are going to just click on this. So sorry for that. You click on this and it's going to take you to this. Now, this is actually the number we need. I'm going to go back and re-explain so we don't get confused. Now, this, when you get, let me just start, go back. When you open the interface and you get the specific work you want to work with, the research you want to work with, when you click on it, now you just go down. Like I said initially, you click on more. Where there are samples, you click on more and you choose which of the replicates you want to work with. This is the control, this is the treatment, control, treatment. So like I said, I will use one control and one treated. So I've clicked on the treated, the control. Now we come down. Here you see the SRA. You are going to click on it. We are not copying it. You click on it. You go down. Now, this is the number we have tried the run that has S arrow arrow. That is the actual number we are going to be using. So this is the number you will copy. So sorry for that error. So you come here, you come to your use galaxy, this is the same thing, get data, download and extra read in FASTQ. So that number you copied is what you are going to paste. Once you've copied, paste that number, you come here and you click on run. It runs the data. Good. So what happens when you run? The galaxy will help you to determine if it was a single pair end data or if it was a paired end data. Right now, it's just these two are here. It says a list is zero data set. So it's going to determine which is a single or a paired. While this is running, I'm going to get for my treated now. The one that is running is for the control. So let's get for treated. Just click on one, come down, click on SRA, and we come and we copy the run number. So still come to get data. Here we come to get data, you come to download and extract read in FASTQ, and you paste the number too. Okay, so and we still run it. So both are running right now. Both are running right now, and we'll just wait for it to finish running. We'll wait for it to finish running. Okay, so both data are finished. They are finished the analysis, and they are both paired in data. You can see a list with one pair. You open it, this is what it shows, the forward and the reverse. It shows the forward and the reverse for both of them. So they are both paired ends. This is it. They are both paired ends data. So before we go into the analysis, we are going to do something. We are going to tag these our data. Now, why do you tag them? Because as you go along with your work, these data are going to be, you are going to do a lot of analysis. So you can know which exact one you are working with, because a lot of data results will be produced. So know the exact one you are working with, we are going to add tags to them. So for me, I'm going to add GO arrow global outreach Vivo, so I can know the one I'm working with. Okay, before you do that, you please always remember to push hashtag. We put hashtag. If you don't put hashtag, you could see what it was showing me before. It's not a valid tag. So until you put your hashtag, it will not be valid. So always remember to put hashtag. And just click on plus and save. Okay, since I have not saved it, do that again. Okay, I think the issue is because I use capital letter, so let's use small letter. Okay, then let's remove the spacing. Let's see if the spacing is what is wrong. Good. So the spacing was the issue. So I've added it as a tag. So always remember to click on save. If you don't save it, it won't work. Now to come back, when you click on it, it won't work. Save. Okay, good. It has worked. So this is the tag for this. Um, and also, I'm coming. That tag, I'm going to rename this tag because I want to know, to be able to know my paired data and my unpaired and my treated and non-treated. So this one is actually the treated. So I'm going to put a capital letter C to show it is treated. So, okay. So this with the capital letter C shows is the treated. And let's come to this. We do the same thing. We add tag, hashtag, zero arrow, if we go, and with a capital letter C to show that it is that is control. So good, my tags have been added. So you see, for the ones that put a tag, it is. So the step now is to do the quality check. And to do quality check, we are going to use a tool called the fast queue. we're going to use a tool called FastQC. Now, this tool is going to be used for checking our data to see how good, like I explained in the first video, how good quality check is done, used to see if it has low quality reads, if it has contaminants of adapters, and a whole lot of others. So the tool being used when you come to your toolbar is the FastQC. Now, this is the tool that we're going to use. So you are going to have to select which of the data, if it's a single, a multiple. So what do? You just come and you click. Good. This is the one we're going to work with control. Now you see why it was labeled at the first instance, because if it wasn't labeled, it wasn't tagged, I wouldn't know which data I'm working with at the particular moment. So we go with control. Okay. So this is we're going first with control, and all these others you just leave them blank. Every other thing you leave it blank. And after doing that, you just run. Good. So it is running. So let's do for the treated. We've done for control. Let's do for the treated and run. Good. So both are running now, and we will just wait. Now you see why naming is important. You can see that those with control has their tag. I can know which data I'm actually working with at the moment. It actually the tags help you to know your data set that you are working with. So we'll just wait for both of them to finish running. Okay, so both data are finished running. You can see that it creates, now this is how it does. It creates two data sets. It creates the web page and the raw data. Now the web page is like a graphical visual. Now let's open it. This is the forward. So this is it gives you a view of everything. This is the encoder, the total number of bases, the total number of sequence, the GC content, the sequence length. And so it shows you how much, what quality it is. Now, what we are going to do next, I usually do this when I'm working with multiple data at the same time. We are going to use something called multi-QC. Multi-QC is like a collector. It helps you add everything you've done, all these analysis you've done, it brings it together so you can view everything at once. So on your tool, you just write okay, good. This is multi-QC. So it's an aggregate. It aggregates the results into a single report. It helps you aggregate them into a single report so that you can view all your reports as one. So here you select which tool was used to create the report, and the tool that was used was FastQC. So we're going to impute it. Now, which type of FastQC are you imputing? Is it the raw or the web? And it is the raw that we are going to be using. So we select. Just come and select. Now, like I said, we're using the raw, so this is the one we're going to select. Just take these two. These two are together. This is the forward and the reverse. You pick them together and you select. Also, you also come. Now we're doing this for both, for the treated and control. You pick up, oh sorry, I did something. Okay, let's come back again. Raw data, you select it. Then for the control, the treated, I mean, you also select. Just make sure. I want to make sure it's actually for the control raw data, that is 14, data 8 and 7. Okay. And for the is data 6 and 5. Okay, so let's go back and see data 8, 7, and 6 and 5. Good. So these are the ones that have been selected. So you can put an image, a report title, whatever. Just all these others are not needed. So after you finish selecting, you run. Good. So multiplicity is running, and we will just wait for that to finish running. Good. So our analysis has finished running. The web page, you just click on view and it gives you a visual of everything. Good, this is it. So let's see. Okay, these are the samples, the forward, the GC content, the number of sequence, unique read, duplicate read. Now, what does unique read mean? It means out of those reads, which one did not repeat itself multiple times? That read, the genes in that read. Now, you remember I said the RNA is made up of adenine, thymine, cytosine, and guanine. So when during sequencing, when it cuts it into reads, each of these reads has adenine, thymine, cytosine, guanine. It just continues going. So what it means when it says unique read is that when reading through them, let's say the first read has adenine, thymine, cytosine, adenine, thymine. We still have another one that has adenine, thymine, cytosine, guanine, thymine. Those are duplicates. But when the only reads you could find adenine, thymine, guanine, cytosine, thymine, that is a unique read. So it has given us the percentage of unique reads to duplicate reads. Going down, you see the quality. This is the quality. Now, this this green means it is very good. This yellowish is bit manageable. Then red means it is not good at all. So any data set that you use that is within the green, it has a high quality. If you have some that are within the yellow, like this, all is in the green zone. But if you have maybe from here to this angle that are in the yellow zone, you can also still manage it, but you may have to trim it. But once anyone is in the red zone, you must trim. If it's in the yellow zone, you can decide not to trim. But once it's in the red zone, you must trim it. This is also the same thing. It's also the same thing. It gives you the position of the the base pair content. These are the base pair content. Now this is the GC content. So you see that everything is all together at the same page. You don't have to start looking at each of them individually to know which one is good, which one is not good. It combines all of them together, aggregates all of them together, and just gives you as one report. So that way you are able to work with everything at the same time. Okay, that is for the status report is like the web page is a visual, while the status is just write up. So most times you understand more from the web page. So that will be all for this video. In our next video, I'm going to be showing you how to do trimming. Even though we don't need to trim for this our data set, I'm going to show you how to trim in case your own data set needs trimming. I'm going to teach you how to trim, and we'll continue from there. Thank you very much. See you in the next video. So welcome to the next section of transcriptomics on use Gallup. So in this section, we're going to be treating how to use Primomatic or cut adapt, and how to do mapping and alignment, and we'll continue from there. So like I said in the other video, after doing our multi-QC, we saw that majority of our data sets were of good quality, so that they were of good quality. But I also said that I will teach you how to do Primomatic, how to use Primomatic or cut adapt in case you have a data set that is not of a good quality. So the tools we use is either Primomatic or cut adapt, whichever tool you like. I will use cut adapt. So this is the cut adapt. Now cut adapt, this is the interface for cut adapt. You are going to choose if your collection is a paired end or a paired end collection. Most of the time, just use paired end collection. And now we are going to choose the data we are going to work with. We are going to do control first. This is for control. We're going to do control first. All these others. Now you come here. Yes. No, you come to other read streaming option. Yes. Okay. So what is asking you here is how low do you want your quality to be? Now, if you remember, I said that the red zone, anything, let me open it so that we can actually see it. We'll come back to the cut adapt. Good. So this is anything below 20 is said to be not of good quality. So what do you do when you come to your cut adapt? Let's go to the process again. Control, come down. Other read streaming options. So the quality cut off. What you do is you choose the least quality you want it to cut. So if you want it to be from 20, anything below 20, if you cut it off. You can choose 30, anything below 30, you cut it off, depending on how strict you want to be with your work. For me, I will just put 20. But like I said, you can always choose based on what you want. So this, why it gives you two is for paired and single end. but you can use only this for both paired and single. When you have single, you use only this, but if you have paired, you can use it too. Like you said, for paired, then you can set here a separate policy to apply to with two. That is for maybe for the forward, you want it to have something. For the reverse, you want it to have a difference. You set two. You can set two different quality control, but you can also leave it empty, and this will be used as the borderline for all of them. So after doing that, you come, you just come and you also come here for maximum length. So maximum length too will also be 20. Maximum length will also be 20. This way, any width that is shorter than 20 will be discarded, will not be used. That is what this is for. Any width that is shorter than 20 will not be used. Okay? So after setting all this, you just come and you run. Good. So it is running the for control. So let's do for the treated. Okay. This is for the treated. We do the same thing we did in control. Twenty. Okay. Also 20. Good. So and we run. So it is running and we just wait for it to finish running. Okay, so analysis has finished. If you open it, this is the forward. Now mind you can also use Bioinformatics. You can also use fast QC or multi QC. You after doing cut adapt, you do fast QC again to see how good the quality is now. So after doing fast QC, you still do multi QC to aggregate your analysis together. But since I just showed you this to be able to for you to know how to do use cut adapt in case your own data set is not of good quality. So we won't go through the step of going to do fast QC again and multi QC. But if you did cut adapt because your data was not of a good quality, after doing cut adapt, you are going to do fast QC again and you do multi QC again to get an aggregate. To get an aggregate. So for after that, we're going to go to the next step, which is the alignment. Now, alignment can be done using two type of tools. You can use HiSat. This is it, HiSat, or you can use star, RNA star. RNA star. So depending on which you want to use. I personally like using HiSat. I personally like using HiSat. So this is HiSat. HiSat. So like I said, HiSat is used for alignment. Awareness star can also be used for alignment, but personally, I like using HiSat. So this is HiSat. On opening, this is how HiSAT looks like. Now, HiSAT has a, remember I said that alignment is done with referencing to or mapping to the reference genome. Now, Galaxy has its own inbuilt reference, but you can also get the reference for your data. Now, to get the reference, because I am using a microorganism, which is Vivo coloring, I'll be going through NCBI genome. This is it, NCBI genome. So once you click on NCBI genome, you search for the organism that you want. For me, that is Vivo coloring, and I have to click on genome. Now I just click on search. Sorry, I think I made a mistake in spelling. Good. So for those of you, this is for those using a microorganism. I'm also going to show you how to use, if you are using human sample or rat sample or any other sample. But for microorganisms, it's best to go through NCBI genome. And once you see this, you just click on the assemble number. Good. So if you take you here, now you just click on download. For FASTA is the reference genome and GTF is the annotated file. You also need to download the annotated file. We're going to be using that later on the course of the analysis. So you just click on the one you want. It's always best to rename your your download, you rename it. I've already downloaded mine, so I won't be downloading it again. Now for those haven't who are using human sample, you are going to be using Ensemble. So you go to Ensemble database and you click on Ensemble genome browser. Now this is for those that are using human sample, rat sample, mouse, Drosophila, whichever one you are using. So if you are using human, you click on human, mouse. If you are using Drosophila, you can come here for whichever organisms you are using. This is Drosophila. So I will just use human sample as a guide, but it's the same for all of them. So you just click on the one you want. Now this is the interface it will show you. For the reference genome, you are downloading the FASTA. You click on the FASTA. So after clicking on that, you are going to be downloading this. You are going to click on the cDNA. Once you click on it, it will download your file for you. And for the annotated file, like I said, you are going to be doing that later on. You are going to click on GTF, not GFF, but GTF. You just, and also you come here and you download. Mind you, do not download Ab initio or any other one. You must download the one that shows all. Same for the annotated, the reference genome. Okay, for the reference genome, once you click on cDNA, it will take you to this page. Now you click on all. All. That is the one you are going to download. Do not download the Ab initio, but all. Now let's go back to, like I said, I've already downloaded my reference genome and annotated file. So how do we do that? You come here after downloading it and choose from file. You go to your download because I've downloaded it. These are the This is my annotated and this is my reference. Like I said, always rename so that you know what you are working with. But for those of you that are downloading from Ensemble, you may not have to rename it because it will already help you name it. But for those downloading from NCBI, always rename. So click on this also. So this will show you how fast or how slow it is. When it has finished sending it. So once you've done this, you just click on start and it will start uploading. Good. So both of them have finished uploading. I'll just close and you can see that they have appeared on my history page. So start, I will be able to start my alignment right now. And what do I do? Firstly, search for reference genome. Since I already have my reference genome, I'm going to use a genome from history. And I'll just click on this. So this is my reference genome. I'll select. Okay, this is a quick one for those that will be downloading their reference genome and annotated file from NCBI. Because it comes in a zip file, you will have to extract it. That was the issue I was experiencing just now. I had to go back and extract it so I can be able to use it. But I've extracted it now and I've uploaded it. So this is my file. Now I'm going to paste the data I'm going to be using now. And I didn't show this during the course of the video, but I went and I got an extra data. Remember at the beginning we got sample one control and one treated. So at over the course of the analysis, we are going to need pairs, two pairs of that's a pair of control and a pair of treated. So that means two for control, two for treated. So I just quickly went back and got it. So to work with that, we are going to be doing. So as you can see, I already have them. This is C2, which is treatment 2, and C2, which is control 2, alongside control and treatment 1. So we're going to be doing alignment for all of them. Now you can do alignment for them individually or as a pair. Now for them as a pair, you use paired ends. No, sorry, you use collection, data end collection. Now this is for them as a pair. As a pair, you use paired ends, and for that, you can do it individually, faster file 1, faster file 2. That's you do treatment 1, treatment 2, or you can do it multiple data sets, meaning that you can use this file 1 to do all the treatments, all the treated, and file 2 to do all the control, depending on which one. But to make things faster, I'm going to use this for all the treated. So I'm just going to come here and select. So I'm going to select all the treated. Now, for the for your alignment, you are going to be using for the alignment, you are going to be using the FASTQ file. So for using the original file that you downloaded, not the FASTQ file, but the original file you downloaded. So this is treatment. I'm going to do all the treated file first. So this is the third end for the treated. And I'm just going to select this. Select this. This has been selected. I'm going to come and get the treated 2. Get the third end of it. To treated 2. This is it. And I'm just going to copy this through and select them also. So this is for the treated. All these are the treated. Now the second file 2 will be for the control. So I'm just going to come and get for control the third end for control, which is this, and I just select this 2 and also select for control 2 and I select. Good. So I've selected all for this is for my control and this is for my treated. So they have all been done. Now I can just run. Good. So this is already running. And we're just going to wait for it to finish running. Okay, so I have to quickly go and rerun it because running all of them together at the same time was causing an error, so I had to rerun them individually, but this is them individually. So this is it individually. Now after high search, we're going to go to the next, which is feature count. Now we know that high start is used for alignment. While feature count, we're aligning every of the reads to the reference genome to see how many genes will be aligned. So feature count is trying to count how many genes we are aligned from each read. So feature count. Now for feature count, we're going to need to put in the alignment file. Okay, so let's go. So we're just going to do each of them individually so that we don't encounter any error again of it. So this is it. Control 1, we're just going to do this. You leave it as unstranded. Now for the annotated file, we're just using from your history because like I said, we downloaded the annotated file. So we leave this as exon, gene ID, you leave this on, then you come here, create gene length file, you leave it, turn it to yes. Then next, you're going to come to, does the input have a read pair? Yes, it does. So you, yes, paired n and count them as a single file. What this means is, you know, for each of these samples and each of these reports, it is the forward and reverse. So what we're telling you to do is to read the forward and reverse together as one. So instead of reading control 1 forward forward, control one, reverse. It's going to read forward and reverse, control one together. So that's just what we're going to do for there. Then come down on this place, minimum mapping quality, we're going to change that to a 10 and we're going to run. Now we're just going to do the same thing for all the other files. Okay, so all of them has finished running. You can see feature count for each of them. Now we can actually read it to get on its own or do multi-QC for all of them. So we're just going to run multi-QC for it to aggregate all of them together and show us in one broad sheet so we can see what we've worked with. So let's just go feature count, multiple data sets. Let's just select all the feature count. Now for multi-QC, we're going to be using the summary for all of them. That's what we're going to be using. So the way we can do it is, if you notice each of them shows this, then when you open it, this is the summary. When you open it, it shows you a file. Also, without having to open it so that you can select all of them at once, just look for the summary. Like I said, we're using summary. So this that is shown is what is actually inside here. So we're just going to click on control so we can be able to select as many as possible. We go to the summary. This is the summary for this. This is the summary for this. Mind you, we're selecting 4. So it's 4 data we are going to be selecting. Okay. Okay, so these are the 4 we're going to be using. Just make sure it's these are the 4 we're using. Just want to make sure the 4 that, okay, so click on select. The 4 of them have been selected, 1, 2, 3, 4. So we just run it. So we wait for that to finish running so we can see the aligned, how many were aligned, how much it was aligned. We'll just be able to see everything. It will give us a visual of what we are working with. Okay, so let's just quickly view that. Okay, so to view, now we can see for each of them, these are the 4 that we did, and we see that 4.1% of each of this was aligned, 3.8. So let's just go down and see. Okay, this is for treatment 2, control 2, treatment 1, control 1. It arranges it according to as we gave it to it. Okay, so for control 1, we see that 3,996 was assigned or aligned. The others were not. The alignment quality was there, and this is just giving us a summary of everything that was aligned. So feature count, after doing feature count, next we are going to go to is the differential expression or normalization. Now, like I said, for differential expression, these genes that were aligned, we want to actually see what they actually express. What do they express? What do they actually do? We know that these genes have been aligned and they're actually there. Like these genes actually represent something and they actually are active. Now we want to see which of these genes, what are these genes used for and how active they are. Now to do that, we're going to do something. We're going to create a new history. You just come to the top of your history and you create a new history. Now on creating a new history, once you click on it, a new history will come up. I'm coming. Okay. So you are just going to quickly rename that so that you know what you are working with. And I'm going to name mine Global Outreach 2. Okay, so I've saved that. Good. So after naming it, now you are going to come to this your setting. All the feature counts that you used, all the feature counts that you did, you are going to export them here. And how do you do that? You are going to use copy data set. Remember, you come here. Good. So now there are two: source history and destination history. Source history is where you are getting your data from, and destination history is where you are taking your data to. So our destination history is global outreach 2, while our source history is global outreach. Okay, so from the global outreach, you're just going to come to where all your feature count is. Now, mind you, for differential expression, you use the count. If you notice, your feature count brings out 3 results: your count, summary, and feature length. For multi-QC, you are using the summary, but for differential expression, you use the count. So we're going to copy the count of each of them. Just click on all the feature count of collection count, the result with count. Just click on all of them. Good. So after doing that, just come up and copy history item. And all of them are going to be present here. You can see for treatment 2, for control 2, treatment 1, and control 1. They are all present. Now, the tool used for differential expression is DE-SEQ2. DE, differential expression sequencer 2. That is what we use for differential expression. So how do we do this? Firstly, you come here, you select database per pair. Now you have your factors. How do you work with your factors? This was the reason why I told you I went ahead and got an extra. If you remember at the beginning of the video, we got one for treatment, one for control, but I told you I went ahead and got an extra one for treatment because during differential expression, the minimum samples you can run for differential expression is 2. The minimum you can run is 2. So if you know you are going to be doing differential expression, the minimum you can work with is 2. You can work with up to 5, 6 for differential expression on Galax. But the minimum you can work with is 2. So firstly, we come to factor. In factor, we're going to name this as, you can name this as whatever you want to. It's optional, but you can name it, like if you see here, effect of drugs or cancer markers, you can name it what you want what your research is about, what your research was about, and all that. That can be the name. Now, if I want to name it according to what my research was about, I will come to where I got my samples from and see what the research was about here. If we look at it, the research is a lot. So what I just do, what I will just say, or what I do, if I have a research in mind, I name it the research I'm doing. But for this section, I'm just going to name it as GOT, Global Outreach Transcriptomics. I'll just name it as that. You can name it whatever you want to name it. This factor is like the name of your experiment. That's what you can name it. Now for this factor level is talking about what you are using. So the first, you can name it as control. The samples you are working with, what are they about? Is it the control? Is it the treatment? Or is it disease and not disease, heat and cold? Whatever you name your samples as, whatever samples you are using, that is what your factor level is going to be about. So firstly, we are going to start with control. Okay, so for control, I'm going to select all the samples, all the files that have to do with control, all the results that have to do with control. So I'm just going to come for control, list, then also this. These are the two control. So I'm just going to select them. So this is for control, and I've selected all that has to do with control. Now you go to the next factor level. The next factor level is going to be treated. And I'm going to select all that has to do with treated. So treated 1, treated 2. So access them. Okay, so this is control and treated. Now coming down, you just have to do some extra things. If I have headers, you click on yes, you leave this as it is. Now advanced options, you come down and go to use beta prior to change it to a yes. For output options, generate plots for visualizing the analysis results. You select that, then output normalized count. These are the two outputs that you need. Once you've done this, you run your samples, and we just wait for that to run. Okay, so our analysis has finished running. Now we have 3 analyses: the results file, the plot, and the normalized count file. What we really want to see is the plot. Okay, the plot will give us a pictorial view of what we want to do. So this is it, treated as yellow and controlled as. So what it shows is that it gives us like a graph or like a heat map. This is the heat map. This is the dispersed or scattered plot. This is the histogram. This is also a scattered plot. So it gives, this is for the normalized count. This is for the p-value. This is for dispersed estimation. Sorry about that. This is for dispersed estimation. This is for the sample to sample distance. Why this is also for the p-value. This is the also for the the samples distance. Okay. So whichever one, you have the options to actually save. This is to save it, to download it into your system and use it for your reports. Now for the heat map, is showing between the feature count Now let's use the histogram. Okay, let's use this. For this, it's showing that because this gives us a definite of the treated. For the treated at this value, the treated is expressed. It has a variance and it's expressed, but at this value, this is where the control. These are the control is downward. Yeah, that's the value I was looking for. The what I was looking for. Now in expression, we have the downward and the upward expression. So for the control is the downward, while the treated is upward. Meaning, what does it mean to be downward and upward? Downward means at that moment it wasn't expressed. So in this condition, this this particular genes at this particular levels, at this particular distance was not expressed. But at this distance, it was expressed in the treated. This is what it means. So after this, differential expression, your transcriptomics analysis has all been done. You save this. You just save this and it automatically downloads. Everything automatically downloads to your system and you can start writing your reports. So I hope this has given you a good insight into transcriptomics on Use Galaxy. Thank you so much for staying with me.